Hi, my name is Milena, and I'm a graduate student at University of Chicago. I work with Arvin Murugan along with Sepa Kuhn and Stefano Alesina. And I am talking about microbial community growth patterns and how this impacts robustness, but more specifically, how minimizing co-growth is a unifying principle for robust communities. So in this work, we're considering communities that are transitioning between growth phases and growth dispersal cycles, and ultimately these communities that have some sort of stable composition of multiple species in coexistence. Within each of these cycles, you can consider the growth dynamics, and these growth dynamics will be determined by a variety of context-dependent interactions. This can be due to the excretion and depletion of nutrients, as well as toxins. It can be due to spatial structuring of sorts, and any number of other factors. And for a specific system, as we've heard in a lot of talks throughout this week, we can really try to tease apart all of the details of this. So let's say, this particular set of growth curves that I've shown you is happening due to the green population being a primary degrader of some complex sugar. Maybe it secretes something that's in a toxin to itself that decreases its growth rates, creates a new niche for the yellow to grow, and finally the blue swims out for some reason, creating spatial structure. These stories often end up being very complex and rely on the incredibly careful experimentation that many of you do. And while we can take away certain pieces of them and translate that to other scenarios, they're often not as translatable across multiple systems. So in this work, what I'm going to argue is that across multiple models, if we extract away the mechanistic details, what really matters is just how much these growth curves are overlapping with one another. And that level of co-growth is going to determine how robust these communities are. I'll show you plots like this for multiple models, in which a single point on this scatter is going to signify one community with a particular set of parameters defining the interactions within a particular universe of interactions. For communities such as the one on the far left, you will have situations in which growth is more co-localized in time. In other words, there's more co-growth, and these communities will be less robust.
So robust means resistant to perturbations.
Yes, I'm about to get to the definition of robustness, but it's time scales before sort of this community diverges away from its stable composition. For communities in which the growth is more separated in time, we'll find that these are more robust. Before jumping into my work, which is grounded in simulations and synthetic data, I want to bring attention to the fact that this is not just a mathematical curiosity, that we're not just thinking about sort of these complicated growth curves because we can construct them in silico, but actually we end up seeing this sort of successional or sequential growth in natural systems as well. So this is probably a paper that many of you have read and seen before. It comes out of Otto Cordera's group, and what they did was they took seawater and they looked at the sequential degradation of chitin particles. And this is similar in two major ways to the kinds of simulations that I'm running. One is, as you can see in the scheme, we're considering these scenarios in which you have chitin particles which are populated by a particular community. That community degrades the particle, and then it'll disperse some representation of that community to a new particle, just like the same kinds of growth dispersal cycles that I'm simulating. You can also see from this heat map, which is showing abundance as a function of time for different taxa, that you have this development over time of the environment, where first you have your primary degraders. They're going to excrete some additional metabolites, which prepares the environment for the next stage of metabolism along this cascade, just in the same way that we see separation of growth in time. So to ensure that we're on the same page, as your question was, I want to define what exactly I'm talking about in terms of robustness. So we're defining robustness as the number of cycles that until the sort of stable coexistence disappears under the pressures of stochasticity. You may have other ways to quantify robustness. However, we think that our results should be qualitatively the same, so long as you're considering metrics of robustness, which are grounded in sort of the time periods for your system to deviate away from sort of this stable state under stochastic pressures. So when I'm talking about stochasticity here, I'm talking about demographic noise. In other words, if you can see from the first case, which is starting at our stable composition, rather than having a perfectly reliable representation of that community in the next stage, we end up having some noise. And this is noise in the fraction of the different populations to one another. So, we ended the first cycle with 1 blue to 2 green to 2 yellow, but then the next cycle was seeded with 2 blue to 2 green to 1 yellow. I also want you to note that we're considering things on two different timescales. Co-growth we're considering within the timescale of 1 growth cycle. But this robustness we're considering then over a longer timescale of how many of these cycles until we have this collapse of diversity. Okay. So I want to jump into sort of examples of particular experimental systems that we've run. So the first mechanistic model that we considered was this case in which you have complex spatial structure as a mechanism for species coexistence. This is a paper that was published a few years ago out of Sandor Tan's group. In this experimental system, you have 2 species that are inoculated in the center. And then there's this trade-off that they find, which enables coexistence between how well you can grow locally on the nutrient and how well you chemotax out. So we have this green species, which is going to grow in the center. It's a very good grower, but not as good of a chemotaxer. It's going to deplete your nutrients, creating this gradient that both of the species in theory can swim along. However, the blue will grow out to the exterior and establish its own spatial niche. Growth in the end, however, is not only separated in space, but it's also actually separated in time. And it's this idea that this complex mechanism of spatial structuring is what's actually then structuring also the growth in time. So if you consider then you do a sweep over parameters, being able to tune these 2 things of the growth rates, mu1 and mu2, and the chemotactic affinity rates, chi1 and chi2, we get this really complicated region of coexistence in parameter space. The complex structure of this region makes it difficult to predict how small local changes in both the physiology of the system as well as the physical properties of the system, whether that be the swimming, the chemotaxis, the growth, or the diffusion, will affect the stability of the system under fluctuations. If we think, however, from this perspective of co-growth, we can see that now we can look at how co-growth is just changing as a function of these parameters. So when we're in sort of the interior of these coexistence regions, we have less co-growth, the lighter colors. When you're approaching that boundary, you start to have more co-growth, the darker colors. But we're actually not just interested in this binary sort of assessment of coexistence, we're rather interested in this dynamic assessment of robustness over multiple cycles. So what we've done is in silico, we've run these experiments. We inoculate a plate, we let it grow, we dilute it over and over again to find the stable composition, and then do the same thing, but now starting at that stable composition, introduce fluctuations and measure how many cycles it takes until there's death of one of the 2 species. In this case, the lifetime would be 2 cycles. So we can run this calculation for the same ensemble of communities that I showed you before in this complex coexistence space. And we find this really striking log linear relationship between our metric of co-growth and our metric of robustness, lifetime. Here in the scatter, a single point is telling you one community that's defined by a particular set of growth rates and chemotactic affinity constants. And if you pull out, let's say, the case of longest lifetime and least co-growth, you can see that the green is growing really well locally, but it doesn't swim out that much. And the swimming out then establishes this sort of secondary bump of growth in time for the blue species. In contrast, for this case of less co-growth, the growth curves are nearly overlapping. Yeah.
Milena, perhaps you'll describe this, but why is a larger number on the x-axis less co-growth? Sorry, that's a bit confusing.
Yeah. So
maybe you'll explain it.
IPA. I don't know. I find this hard to IPA.
Yeah, so this is a metric that is drawn out of basically looking at the ratio. So the way that we're calculating co-growth is we are looking at the ratio of growth rates to one another. It's connected to an metric of invasibility. that was previously published out of Terry Hua's group from Avanish Narla. I'm happy to write down the explicit equation if that would be useful. I wasn't sure that the full derivation was within sort of the scope of time, but that's why this number gets larger as you end up having sort of more, if you think of it as the ratio of, let's say, species A to species B growth, the more that these are different, the larger that number will end up being. And the minimum is because this is sort of an asymmetric measurement where you can look at the advantage of A over B versus the advantage of B over A. And what matters is the smaller of those two numbers.
You're not looking at the overlap between the growth curves in time or space or something like that, right?
Yeah, I mean, it's something which is more like the integral over time, somewhat rescaled, of like the rate of A over the rate of B, 1 over the total biomass, where this is time rescaled as biomass growth. But yeah, so it's this ratio, and the IAB versus the IBA has to do with just like whether you're considering A over B or B over A. So these numbers will be slightly different due to asymmetries. Yeah. Okay, so spatial structure is a good example for demonstrating how we can sort of abstract away the complexity of these complicated PDEs into an overarching phenomenological statement that has nothing to do with space, but we are actually interested in demonstrating this across multiple mechanisms. And so the second piece of work is a piece that I actually do not have fully done yet. However, we're interested in looking at how, on a backdrop of cross-feeding inspired by generalists in denitrification, if you add, in addition to that, toxicity of one of the metabolites, how that's then going to separate, again, work as a mechanism for separating growth in time. And then similarly, we would do a study of the community lifetime. So I showed to you this observation in one particular mechanistic model, but this is actually tied directly to something that we would expect based on mathematical theories. And the mathematical underpinning linking community lifetime to co-growth is ultimately comes down to mapping our growth dispersal cycles back to a random walk with some restorative force. So here, what I'm showing you is I've abstracted away those dynamics within one growth cycle. And instead, we're just looking at how the community composition changes over one growth cycle as a function of the initial community composition. So we're measuring community composition as just now the ratio of abundances of the two species. And here, this you may have seen in other sorts of dynamical systems analysis, but that point at which it crosses the x-axis is going to be our stable composition. Points to the left of that will be forced back in the positive direction because you can see that the change is positive. Points to the right of that will be forced back to the negative direction because the change is negative. And our stochasticity is effectively taking a random walk away from that steady state and moving us up in energy. So here now, right, I'm showing you energy, which is simply our usual definition, which is minus the integral of that force. And despite stability, fluctuations can build up and drive the system towards one of these extinction boundaries. And the fact that this is an absorbing boundary is relevant because, in biology, we can think of this is reaching points at which you have an extreme ratio of one species to the other. And in that next dilution or dispersal step, you would effectively lose that species. So provided these boundaries are far enough out, you can estimate that in that next dilution stage, you have the loss of that species. So physics and thermodynamics will tell us that what ultimately matters to describe the characteristic time to reach that boundary is simply the height of the energy at that boundary. And for mathematical derivations that I won't get into right now, but I'm happy to talk about offline, it turns out that this height actually directly has to go back to this metric of co-growth that we have written. And so we end up with this strong tie back between less co-growth is going to give you longer lifetimes. So I showed you a link between co-growth and community lifetime in a specific two-species mechanistic model. And then I showed you the mathematical theory that predicts this relationship. However, this framework actually generalizes to many species and to many mechanisms, as we can show from a mechanism agnostic model. So to show this, I built a model in which we consider now just random matrices, where the rows here are going to be species, the columns are telling you bin rescaled time, and a single entry is denoting the per capita growth rate for that species in that bin. Once again, I build an ensemble of many communities. Each of these matrices signifies a community that has full coexistence of all 5 species, and I ask what then is the robustness or the community lifetime for those communities under fluctuations. And we again see this strong relationship where less co-growth on the x-axis is going to correspond to longer lifetimes. And from sort of qualitatively, we can see this as well, where that point with the triangle, upward triangle, you have much more shared growth in a particular rescaled time bin versus in the case here on the for the diamond, you have almost a near diagonal, just almost the identity matrix, in which you have really strong growth of just one species in each bin. So in summary, I started with this question of across mechanistic models, what properties of communities improve robustness under stochastic pressures? And I argued for you that through a variety of context-dependent interactions, you will end up structuring growth in time. And it's this structured growth in time where growth will be separated that improves the robustness. I showed this to you in 2 instances, one which was a specific 2-species mechanistic model in which we had complex spatial patterning to separate growth in time, and one which was a 5-species mechanism agnostic model. So to leave you with one final question to ponder, I want to state a consequence, actually, of what happens when you drive a system too far to the extreme degrees of separated growth in time. So in our definition of robustness, we were considering the time scale with which it takes for a community to reach a point of an extinction event. But we don't consider what then happens with the dynamics of that community after the loss of that species. In minimizing co-growth, you effectively create a reliance on a series of species that are achieving a sequence of events. And if you begin to then remove one of those species, you can cause stalling because the other species will have to make up for whatever task that species was doing. So we started to look at this in the context of our mechanism agnostic model, in which here on the y-axis, I'm showing you the ratio of the timescale when you leave one of these species out versus if you leave the full 5 species system. And for instances like these ones, in which you have sort of one species that's really doing all of the work in a particular time bit, it's going to stall because it's going to take much longer for the remaining species to do that job. So we looked at this, we've started thinking about this in a context of arrested development, and we're curious to see how this may apply or not to biological models in addition to our mechanism agnostic model. So with that, I want to say thank you to a variety of people, my mentors as well as collaborators, and funding sources and affiliations.
From Chinese.
Thank you, very nice. When you show the correlation between 1 over co-growth, I think, and the lifetime, it seems that the deviations to that correlation, after like slide 32, yeah, it seems like the deviation to that correlation increases, right?
Yeah.
And it's kind of symmetric relative to that correlation. Do you know what those communities mean? Like the
yeah, so this like linear relationship is sort of after a variety of approximations, and so really we would expect that it should be sort of anything below a line, and that's because I'll try to pull something up from one of the hidden slides. So, oh no. Sorry, I have to unhide the slide, I guess, in order to be able to present it. Okay. So it comes down to the fact that we're approximating this region as a rectangle, which will be true the more sharp that this curve is. But your rectangle will end up being somewhat of an overestimation. So we do expect that there may be some points underneath that curve, depending on the specific dynamics of what's defining how sharp this boundary is. Hopefully that answers your question.
Because you have deviation.
You have deviations
Yeah.
from the correlation to both sides, let's say, like an anti-correlation.
So I guess I would say then that the line is like this.
I see.
Ones are below.
Okay.
Yeah.
Thank you.
Hi, Milena, very nice. So maybe I didn't understand correctly, but a question. Can you also see maybe a relation between the diversity and the number of species with the overlapping growth? Meaning that somehow this overlapping growth can give you an estimation of the temporal niches of your system. So you could try somehow to also count how many temporal niches you have. And this is related to the number of species that you can pack. It's something that you can do in this formalism, or I didn't understand anything.
So I think that this ties into more work that's kind of similar to what is coming out of the paper that was published by Avanish Narla, as well as by Sergey. He has some stuff which is looking more at how you can pack these temporal niches. I think what we're really looking at here is how we're not necessarily focusing on this concept of creating temporal niches. We're looking at how like all of these diverse biological mechanisms that we know that are creating niches of a variety of sorts, whether it be spatial or metabolic, are really just different ways of then structuring growth in time. So we weren't necessarily considering really diversity in this context, but I would reference you towards Avanish's paper is in PNAS. I think it came out maybe 2023 or 2024. And then Sergey also has some work looking specifically at packing temporal niches in order to see diversity in a variety of other factors. Sorry, I was looking around trying to find you, and then you're right in front of me.
Yeah, so thank you. But I'm a bit confused about one point, right? So if I think about, like, say, the mutualistic system, right? They grow synchronously. So like because of mutual dependence, they grow synchronously all the time. And if you dilute them down, it doesn't matter like whether the fluctuation, how big the fluctuation is, as long as it's above some minimal total density, it will grow back up. So I would say that has the like, maybe I understood it wrong, but it seems to me that is a system of complete co-growth.
Yeah.
Right. But it's a highly robust.
Yeah. So maybe hopefully I'm understanding your point correctly, but I guess what I would say is that we're not saying that co-growth is necessarily required for these highly stable systems in terms of having a stable coexistence. But I think so one of the models that I have run sort of simulations for is really just a simple cross-feeding model in which you can then have sort of a similar thing where you just need both nutrients, but it doesn't necessarily matter as much in time. But from this definition of robustness in which you're considering sort of whether I guess maybe it comes down to then the fact that a single species cannot survive on its own. I don't know. I think maybe I will have to think about that a little bit more before I give a detailed answer. Yeah. But it feels like it comes down to maybe slightly different definitions of what we're talking about in terms of robustness and stability. But I would.
But then you need to add a qualifier,
Yeah.
right? Minimizing co-growth in species that can, that are not obligatory dependent.
Yeah.
Otherwise, it just doesn't sound right.
Okay. Thank you.
I mean, maybe getting to Wenying's question, is this basically could I interpret your results as basically saying that on like a single like niche dimension, big air quotes, no one quote me on that, but along like a single niche, I can like create this coexistence by like separating in time. But in like Wenying's case, it's just two completely separate niches, right? Like they're just occupying completely non-overlapping niches. And so could I sort of abstract this to some like multi-dimensional space where things are sort of like going along a few different niche axes, but then the coordination in time is important along each axis to like allow co-growth along that exact like that axis itself. And so there's some sort of complicated space allowing for coexistence of many species.
That makes sense to me.
Other questions?
Yeah, because
Was your model agnostic framework still in the serial dilution setup?
Yes.
Did you explore having like a wider pool of species distribution for like the whole glass jar of seawater, going back to the experimental example? Because in this case, are the initial conditions correlated then between the serial dilutions?
Sorry, could you
Sorry.
rephrase that maybe? I'm not sure I'm entirely understanding.
I imagine in the experimental analog you gave at the beginning that like each particle is somewhat independent in their initial colonizations. But in the serial dilution, there is a fixed dependence between each dilution, right?
Meaning you're saying like bringing back this idea that if you allowed for some sort of invasion from a broader species pool or something like that.
Yeah.
Yeah, I mean, I think in our case, we were considering sort of so I can say very specifically for the way that these growth matrices were created, we actually were pulling from a larger species pool. And then we selected just like the 5 that would coexist. And then that defines sort of the stable coexistence composition. And then you were looking at sort of the robustness of that under fluctuations. So I think maybe in that sense, it's sort of saying that if you were to look at a particular stable community and start from that as a point and then see how that community evolves. But it is sort of an important component that we're not considering invasions from other species outside of the species that are included in the system, or even from species that go extinct over the course of these fluctuations.
Thank you.